everybody and welcome back to my channel. So today's video is the final one for my protein synthesis series, and this is for OCR, AS and A Level Biology A and B. So our first specification points are for all of the OCR, AS and A Level specifications. So number one is the nature of the genetic code, triplet code, and how a gene determines structure of proteins, including enzymes, by ordering the sequence of amino acids in a polypeptide. Number two, transcription and translation of genes resulting in the synthesis of polypeptides, to include the roles of RNA polymerase, messenger RNA, transfer RNA, and ribosomal RNA. So for our first specification point, here we have our DNA and we have our gene, and a gene is a section of DNA. So a gene is made up of a sequence of bases, and this is the code that codes for the sequence of amino acids, and it's the order of amino acids that determines the polypeptide chain or protein produced. So our gene is made up of bases, and in DNA these are A, T, G and C, and these bases are arranged in a triplet code, so a sequence of 3 bases codes for a particular amino acid. So here we have TAC, so that codes for this particular amino acid, then CGA coding for another amino acid, TGC coding for a third amino acid, and so on. So now we're going to be discussing protein synthesis, and there are 2 stages. The first stage is transcription, and this occurs in the nucleus of the eukaryotic cell. So we've got our DNA and we've got our gene, and the enzyme called DNA helicase breaks the hydrogen bonds between DNA, and the DNA molecule unwinds. Now make sure to check with your teacher as to whether you need to know about DNA helicase for protein synthesis, as in all the A-level specifications it's only mentioned in DNA replication. So yeah, just see whether you need to know about this. Some of the specifications replace DNA helicase with RNA polymerase for this step as well, or they just say that the hydrogen bonds break and the DNA molecule unwinds. So just check with your teachers on that. So once the bonds are broken and the molecule unwinds, we are left with 2 DNA strands separated. So we've got our sense strand, which is also called the DNA coding strand, and our antisense strand, which is also called the non-coding or the template strand. So it is the template strand that is actually used for copying of the code to mRNA. So we've got our coding DNA strand that has the code of the gene, and then the template strand almost reflects this code onto it, and then the code is copied onto the mRNA. So RNA polymerase actually lines up 3 complementary RNA nucleotides alongside the DNA's template strand, and then hydrogen bonds form between the bases. So just a reminder that the bases for DNA are A, T, C and G, and the bases for RNA are U, A, G and C, so not T but U. And the way they pair up is DNA's A base pairs with U, DNA T base complementary base pairs with A, and those 2 sets are with 2 hydrogen bonds, and then DNA's C base hydrogen bonds with G, and G hydrogen bonds with C, and they have 3 hydrogen bonds. So once the RNA nucleotides are all lined up, hydrogen bonds between the bases break, and RNA polymerase actually joins the mRNA nucleotides, forming the pre-mRNA strand. So after this, transcription begins, and this is in the cytoplasm. So the mRNA moves out of the nucleus and into the cytoplasm, where it attaches to a ribosome, which is the site of protein synthesis. So another molecule called tRNA brings a corresponding amino acid to the mRNA at the ribosome, and its anticodon complementary base pairs with mRNA's codon, and the amino acid corresponds to the code on the mRNA. Then a second tRNA molecule carrying a corresponding amino acid complementary base pairs to the second codon on mRNA with its anticodon. So ribosomes are actually made up of ribosomal RNA, and it's the job of the ribosomal RNA to catalyse the formation of the peptide bond between the 2 amino acids attached to the tRNA molecules. So once the 2 tRNA molecules bring the amino acids, they then form a peptide bond between the amino acids. So this continues until tRNA reaches a stop codon and the polypeptide chain of amino acids detaches from the ribosome. So now we're going to move on to some questions based on that. So our first question for biology A is an anticodon sequence of 5 successive tRNA molecules involved in protein synthesis was analysed and found to have the following percentage base composition: adenine 40, cytosine 27, guanine 13, thymine 0 and uracil 20%. Which row shows the percentage base composition of the template strand of the original DNA molecule? So let's highlight the key words here. So we've got tRNA. So we know tRNA has anticodons. So the anticodons it has are A, C, G, and U, because thymine is 0. And we know that because RNA is made up of these 4 bases. It's not made up of thymine. So we know that our mRNA strand codons complementary base pair with our tRNA. So if we work out what they would be, so adenine A pairs with U, C pairs with G, G pairs with C, and U pairs with A. So our mRNA is going to have the same percentages of this, but for these bases. So then if we work back again, the template strand is going to have then 40% A, 27% C, 13% G, but then we need to remember, because it's DNA, it's going to have 20% for thymine for T, not 20% for uracil. So if we have a look at our answers, we can see that we know that C and D are going to be wrong because they include uracil. So that's not correct, so we can get rid of those. And then we've got B and A. So we worked out that thymine is going to have 20%, and the 20% is here. So that means that A must be our correct answer. Question 2. Pepsin is a protease enzyme with a polypeptide chain containing 327 amino acids. Titin is the largest known protein. It has a polypeptide chain containing at least 92 times more amino acids than pepsin. DNA sequences in genes code for polypeptide molecules such as pepsin and titin. Explain why a process known as transcription is necessary for polypeptide synthesis. So this is asking us about the importance of transcription in protein synthesis and why it actually occurs. So we know that the actual process of transcription is the gene being transcribed or copied into the mRNA molecule. So that's going to be our first point. And the reason that this occurs is because the DNA molecule itself is too large to leave the nucleus. So therefore that's why the smaller mRNA strand is created. And so the RNA, the mRNA strand, is then able to leave the nucleus and go to the ribosome for translation to occur. So we can see our answer here. Question 3. Now this is part 2 of the previous question, and it is to calculate the minimum length of the DNA base sequence required to code for titin. So we have got some key information in the question we want to highlight. So we've got that pepsin has 327 amino acids, and that titin is has at least 92 times more amino acids. 92 times more amino acids. And then it's asked us to calculate the minimum length of the DNA base sequence. So we know that for each amino acid, 3 bases code for it. So therefore our equation is going to be 327, because that's the amount of amino acids pepsin has, and then this is going to be times by 92, because titin has 92 times the amount of amino acids. So we've got our amount of amino acids, so now we need to times that number by 3 in order to work out the number of bases within that amino acid sequence. And our answer for that is therefore going to be, so 327 times 92 times 3 equals 90,252. 90,252. And we can see our answer there. Question 4. In response to infection by rhizobium, bean plant nodule cells produce protein called leg hemoglobin. Researchers wanted to find out more about 3 genes that code for leg hemoglobin. They use RNA interference, RNAi, to inhibit the production of leg hemoglobin using miRNA. They measured the relative transcript level of the leg hemoglobin genes of bean plants treated with miRNA, RNAi plants, and those of untreated bean plants. And the question is, describe how miRNA inhibits the mRNA of the treated plants. So we can obviously say that mRNA is produced during transcription and then it's translated to form this leg hemoglobin protein. So miRNA is going to inhibit it by preventing the translation of mRNA, and this is because it's going to bind to a complementary site. And then the Argonaut protein is going to break or cleave the mRNA strand at a specific point, which will prevent the transcription and translation of it. So there's our answer there. So now we're going to move on to some questions from Biology B papers. So question 5, 33 human blood group systems are known to exist. 2 of these are the ABO blood group system and the HH blood group system. Explain why a person whose blood group is AB expresses both A and B antigens on the surface of their red blood cells. So we know that antigens are in fact proteins. So it's basically asking us how proteins are expressed within the blood group how that proteins are expressed on the surface of these red blood cells. So we can say that the genes of these proteins are transcribed, and then that allows for both antigen proteins to be produced, and we can see our answer there. Question 6. Proteases in the small intestine of an athlete will break down proteins in sports drinks. The amino acids then enter the bloodstream and are transported to muscle cells. In the muscle cells, amino acids are used to synthesize proteins such as myosin. Explain how amino acids are transformed to and taken up by muscle cells and then incorporated into proteins. So obviously the part of this question regarding the transport of amino acids and them being taken up by muscle cells is not part of protein synthesis, not part of the content I've done today. But I've included this question because it's a good question to write about protein synthesis. So if we just focus on the first part of the question, the last part of the question about proteins, so we could say explain how amino acids are incorporated into proteins. So how proteins are formed, basically. So we basically just need to describe the process of transcription and translation. So instead of explaining it all again, if you go back to the slides again, go back in this video, have a look through transcription and translation and try and answer the question for yourself. So we've got our answer for the protein part here. So because it is a style question, you need to remember to write with good English, and when writing about the formation of proteins, you need to reference both transcription, translation, and RNA structure. And then there's some scientific points to include here. So we could talk about the nucleotide bases, A, T, C, and G, talk about the codons on mRNA, the anticodons on tRNA, hydrogen bonding between the bases when mRNA is produced, and also between mRNA and tRNA. Also the unzipping of hydrogen bonds, when the DNA strands are separated, the production of mRNA, formation of phosphodiester bonds between nucleotides, the role of tRNA, obviously the formation of peptide bonds between the amino acids, and extra points about complementary base pairing and protein structures as well. Question 7. The sequence of nucleotides in DNA provides the genetic code for synthesizing proteins. The genetic code can be described as universal because it is the same in almost all organisms. Name and describe other features of the genetic code. So we've talked about two features of the genetic code today. So we're going to go over those two. So the first one is obviously the triplet code. So we know that a sequence of 3 bases codes for 1 amino acid. And also we've talked about how the genetic code is degenerate, and I haven't explicitly said that this is what it's called, but basically that means that there is more than one codon that codes for a single amino acid. So if we have a look at our answer here, so we've got degenerate, we've got triplet code, and then the final thing is that it's actually non-overlapping. So I haven't discussed that, but we can have a look at it now. So basically it's the idea that a sequence of bases is read a triplet at a time, a sequence of 3 bases at a time, so it does not overlap. But I will be doing a video on DNA structure and also RNA structure soon, where we can talk about this in a bit more detail. Question 8. Messenger RNA, mRNA, is the type of nucleic acid that carries the genetic information on DNA from the nucleus to the site of protein synthesis. Unlike DNA, mRNA is relatively unstable and has a short lifespan. So just one advantage of mRNA being relatively unstable with a short lifespan. So we can say that this is an advantage because proteins are not always needed to be produced constantly. So therefore, they're not required. So mRNA doesn't need to be synthesized, so nothing is wasted, basically, as you can see from the answer there. So thank you so much for watching my video. Please give it a big thumbs up if you did find it useful, and comment down below any other video requests you have, whether they're biology related or just general study related. Also, please subscribe to my channel and recommend to any friends that you think would find this really helpful. Also, make sure to have a look at my Instagram page and my Facebook page as well, both Biology with Gracie, and also check out my website where you can access loads more exam questions and revision resources and notes that you may find really helpful. So I will see you in my next video. Bye guys!